Hello, and welcome back to Scanning for Intelligence. We're doing a proficiency review, continuing on with part 2. We're going to talk about the energy that the cell makes, how does it make energy, and as well as the cell cycle. So let's just dive right on in. So effectively we're going to talk about first is glycolysis, then the Krebs cycle, also known as the citric acid cycle, the electron transport chain, which occurs in the mitochondria, and the cell cycle, as well as just a quick overview of lambda-phage and viruses in general. So, first of all, what is a glucose? Glucose is a sugar. It is ubiquitous pretty much for the idea of the central aspect of going from just a molecule into energy. And so what happens is you can have glucose in the closed form and the open form. And in this closed form, that oxygen is this oxygen right there. And so you have essentially this down, up, down, down, or right, left, right aspects of glucose that has different specific functions. And so what happens is you go through glycolysis. You take one of these glucose, you take 2 ATPs, so you attach some phosphates on, you can get some weird intermediates, going from to like 6 glucose phosphate and then to like 6 fructose phosphate, so on and so forth until you get pyruvate. And that is what the molecular structure of pyruvate is. Double bond oxygen, and then you have an oxygens sharing a negative charge. And then, so what that pyruvate can do is it can either go to the mitochondria and make some more energy, or it can just go into lactose and make a little bit less energy theory. But also out of this you can make 4 ATP and 2 NADH. NADH is just another energy carrier that is used in this electron transport chain. So there are a couple of different other ones as well. There is also FADH2, but as we also know in the cell, most typically we energetically use ATP in the cell. And ATP has about 7.3 kcals per mole for shooting out the gamma phosphate. NADH is 52.6, so 53 kcals per mole, and FADH2 can make about 43.4. And these carriers are used in the electron transport chain to create a chemiosmotic gradient. I'll get to that in a little second. So then we go to the Krebs cycle. So pyruvate can go to the mitochondria. This glycolysis, this happens in the cytoplasm of a eukaryotic cell, or as well as the prokaryotic cells. Then we go to the Krebs cycle, or the citric acid cycle, which we take the pyruvate and then we add it on to acetyl coenzyme A, so we can make it to acetyl coenzyme A, and then that gets made into citrate, and then you go around in a loop of just like 12, 13 steps. Maybe it's less, probably is less, but it's too many steps to count. And what happens is when you go through a single run through of the citric acid cycle, you make 3 NADH, 1 FADH2, 1 GTP, and 1 carbon dioxide. So getting through the Krebs cycle, you can get all these guys. And so now that we have a bunch of NADH, we have a bunch of FADH2, we can go into the mitochondria and further do the electron transport chain. So essentially, what we have in the mitochondria is it's the powerhouse of the cell. We have 2 different membranes. We have the outer membrane and the inner membrane. And what happened, why we have this weird wiggly thing, this wiggly bilayer, this wiggly boy, it actually creates a lot of surface area so you can do all this all the time at pretty much all points on this. So what happens is essentially the idea is you have this intermembrane space right here, and then you have the matrix space. And effectively the goal is to take all the positive charges and take it outside into the intermembrane space. So you usually take the protons out, and then what you can do is you chemiosmotic coupling. So essentially osmosis says these hydrogens, these protons, positive charges, should go into this space because it's not as nearly as positively charged. And so you've got a gradient of just concentration as well as coulombic charge. So that is what that chemical ion coupling does. And so by moving that inwards, you can generate enough of a force in order to make ATP. So how do we get just all these protons out into the intermembrane space? And the way you do that is by using NADH, FADH, and complex 1, 2, 3, and 4 in mitochondria. So this power complex of the cell has these 4 complexes, and the first one, the goal is to take NADH and take it into NAD plus, and it also add 2 hydrogens onto quinine. I don't know how to pronounce it, so go ahead and say in the comment section how badly I mispronounced it and make fun of me. I don't care. So this quinine gets oxidized with some hydrogens added to it, and in this entire process of 1, we get 4. We have 4 protons leave. And then with number 2, we make FAD into FADH2, which stays entirely inside the matrix space, and there is no net movement of protons outside. But then we go to 3, we use the quinine from step 1 and the FAD from step 2, and with some cytochromes, that's cyto here means cytochrome, and then we take that and then we take the quinine, reduce it, we also reduce the cytochrome, and we also take the FAD into FADH2 into FAD, and then we have 4 protons move out in total. And then we take those cytochromes here and we release some protons with O2 with oxygen, and then we go and we make some water, and we also move another 4 hydrogens out. And then, now that we have gone through one single step of this, and this constantly happens, we take F, this is the matrix, which once again, this is the matrix, the outside is the intermembrane space, which naturally makes kind of sense. And so these protons will come on in here and join in, and they essentially, what happens is, this is F1, F0 complex, F0, F1 complex, and so you have this large subunit, this large unit, you have this guy rotate around and around and around, and it will shoot out a hydrogen, and at the same time, you will make a ATP by adding on, essentially, some phosphates onto some DDPs, or DNMPs, sorry, my apologies. When you have monophase, so you can make that, and approximately 36 can be theoretically made, but it's only 32 that are actually made by the cell. Check that number, I don't know if that's quite correct, but anyways, that's essentially how you make energy in the cell by using this template as of glucose. So you can go through a lot of different other steps by metabolizing lipids and amino acids, but those are a little more fanciful pathways, so we're just not gonna, we're gonna ignore that, and figured everyone in Proficiency is probably also gonna ignore that as well. So now we have the cell cycle. We can make energy in the cell, but what happens once you have that energy? And the way you do it is you have the cell goes through a growth period. So a cell is first of all birthed, and then it enters the G1 phase. The G1 phase is growth. It essentially just grows and grows, makes the organelles, and makes it so it's like its size, so it can be like a reasonable cell and be a productive member of society and say no to drugs. And essentially, and effectively, most cells actually leave this G1 phase after they grow a little bit, and they go into the G0 phase. And that G0 phase essentially means it's just not going anywhere. It's not going to grow anymore. It's not going to It's not going to undergo mitosis. It's just going to stay there, stay healthy, stay happy, and just do its main job. But sometimes you can also kick it back into G1, and then you enter the S phase. The S phase is the synthesis phase. What does synthesis mean? DNA synthesis. So essentially, you just replicate all the DNA using the DNAP, which was mentioned in the previous review. And so essentially it just reproduces all of the DNA in the cell. And then once you've done that, you go into G2. So you essentially make some more organelles, make some more size, and make sure that everything is right for the time of mitosis, the M phase. And then Once you have that, you can mitosis and come back and create a new cell, and undergo this a lot of times. Now, all of these steps, as well as the steps in mitosis, are run by, they have several different checkpoints alongside this, and it's run by cyclins and cyclin-dependent kinases, CDKs. These guys modulate essentially the stage of life that is inside this cell. So, going out from here, mitosis has a couple of different processes. So one thing to remember is I pooped myself at the carnival. Interphase is this time with G1S and G2, where you spend the entire time essentially in this state where you have 98% essentially. When you are going to undergo mitosis, you 98% of the time are going to be in this interphase state where you're not actively trying to make into two different cells. So interphase, so I just stated interphase, so I pooped myself at the carnival. I pooped myself at the carnival. And so we're just going to walk through all these steps. Pooped, prophase. Essentially where the DNA condenses into chromosomes. You know, the guys that look like this. What we see? So we start off in interphase with the nucleus kind of just randomly oriented. Then you go to prophase, where you condense them. So you got these condensed chromatids, and then you go on into prometaphase. Oh, sorry, one thing also that happens is you form some spindle poles, some centromeres at the end of two sides of the chromosome. And these guys will come into handy in a little bit. Now, we go to prometaphase, where this envelope, the nucleus essentially disappears, the boundary between it, and the spindle poles attached. So what happens is you have like two chromosomes like this, and then you've got spindle pole and centromere over here. They're both attached, and so they're attached now to the connectochore at the center, or the centromere, centrosome, yeah, at the centromere of each of these guys. Then we go into metaphase, where we take the chromosomes and we actually align them up. Boop, boop. So they're all aligned now in the center of the cell. And then we go into anaphase. So we take that and we just pull them apart. And so that's anaphase. So I pooped P/M T. Oh, sorry, anaphase A. And then we go with telophase. So essentially, it's just like what happens when you're between two steps of. Essentially, we've got this now. So essentially, we just pretty much recreate two cells on the opposite sides, where you decondense the chromosomes, and then you have cytokinesis, where you split them apart, and you just make two different cells. So, I pooped myself at the carnival. Interphase, prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. So once again, you start off completely uncondensed in this regime of G1S and G2. You go and then you, in the prophase, you condense them and you make some spindle poles. In prometaphase, you attach those spindle poles and the nuclear envelope decondenses. In metaphase, you line it up in the center. Anaphase, you pull apart. Telophase, you reform the cells essentially as two different ones. In cytokinesis, you split it off. So that, now I need to talk to you about the lambdaphages and viruses. So lambdaphage is essentially a bacteria that is infected with a virus. And one thing to note is that viruses do not jump domains. So like if a bacteria has a virus, it's not going to affect me. But say a monkey has a virus, it might affect me, which as we know has a very high concern with the livestock in the current climate with the coronaviruses possible to spread. So there are two different types of virus essentially. You got a lytic versus a lysogenic. Lytic virus, it's all cytosolic. So essentially the virus stays inside the cytosol and makes its own proteins and reforms its own viruses, grabbing on and releases. And so it does this a bunch of times until essentially the cell fills up with viruses and the cell bursts and releases all these viruses. Typically, lysis does not actually occur, there's just a gradual, slow cell death. However, you can get a just full burst of lysis and release all these viruses all at once. But this does not enter the genome in any way. But we have a lysogenic virus. A lysogenic virus does a lot of different things, where in particular it integrates into the cell's genome. So it enters the genome and that cell is now screwed essentially. It now will always produce the viral proteome, and because it has the viral genome inside its own DNA. This is special viruses that cause like cancer in humans as well, like HPV, herpes, HIV. These are lysogenic viruses that integrate into the DNA of the host, whereas lytic viruses, such as coronaviruses, do not integrate into the DNA of the cell itself. So that is all for the proficiency review. We were able to go through what glucose is, about the steps of glucose to pyruvate, and then how pyruvate gets made and into the citric acid cycle, and we get some second carriers of this of the electron transport chain for NADH and FADH2, which are then used in the electron transport chain. They get transported using some different symporters and antsporters, which will be covered next time in the proficiency review. And then all these guys have their own purpose of moving high protons out, and then they go through this F0, F1 complex and make ATP. Then we have a cell cycle where I just went over this a bunch of times, and we have a virus. And viruses are bad. So that's all I wanted to cover. Thank you very much. I am Mr. Johnny Hopkins. I'll see you next time.